This is a special episode of the Immunology Podcast, Immunology 2024, Day 2. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rowd. Welcome back to the Immunology Podcast. We have conversations with immunologists. Today we're back with another special episode to discuss the highlights from the Immunology 2024 annual meeting taking place in Chicago. If you're at the meeting, make sure to drop by the Immunology Podcast booth, number 708, in the exhibit hall to meet the team and win some prizes. You can also chat with us about your research and find out how you could be featured on a future episode of the podcast. Today, we also hosted our very first episode recorded in front of a live audience with guests from Seismic Therapeutic. If you didn't make it to the show, the episode will air on May 21st. We'll get more into our experience with that in just a bit. We'll also be discussing some of the most interesting research we've seen presented over the past 24 hours of the meeting. So if you were in another session or weren't able to attend the meeting, we've got you covered. We're going to kick things off in just a second here, but before we get to that,
attending the conferences can generate big ideas that could lead to the next immunology breakthrough, and STEMCELL would like to help you out cover the cost. Come and meet with STEMCELL at booth 603 to enter for a chance to win one of our 4 awards of $500 towards your travel expenses for AI 2024 in Chicago. This contest is applicable for individuals attending Immunology 2024 and closes on May 7th.
all right, we got a full day in.
Oh, what a day. What a day, Jason. Are you happy about today?
It was. The 4th was with me.
The 4th was with you, with us all, thanks to you.
Yeah, I was in Jedi robes.
Yes. So for those who did not attend today's live show, ever first live show, we had Jason honor the day with a Jedi robe. So he actually walked on stage wearing a Jedi robe, which, I think he had a lot of support from the audience. And that was his way of saying, I'm ready to take on this task.
I need to clear my mind, be one with the Force.
Yeah, one with the Force you must be.
Indeed. And we got to see a bunch of talks too today as well.
Indeed.
So I'm going to surprise you with the first one I went to, the first session. It's going to be a real shocker. It was microbial and environmental influences on the gut.
No way.
Oh, we went there.
Yes way.
I think today is the only gut day, though, so we'll get to other parts of the body tomorrow. But from this session, I had 2 talks I wanted to highlight. The first one was from Jessica E. Kennison White from Brigham and Women's, and it's identification of environmental factors that promote intestinal inflammation. So she used a zebrafish model of colitis called TNBS, which is interesting. And I will say, she said this model in mice is T-cell mediated because TNBS is a hapten, and she was saying that means it's T-cell mediated in larval zebrafish who are transparent. That's why they're fun to use. But zebrafish don't have functional T-cells until many more weeks after they aren't transparent anymore. So I didn't get to ask that question, but it's clearly working, and TNBS colitis in zebrafish is a thing. So it may not be T-cell mediated, but I don't think that actually matters in the same way they were getting at. So anyways, they did a drug screen of known toxins from the government and used that to see if they could screen things or making colitis better or worse. And they found 13 worsening and 4 improving toxins, and others that killed them outright or didn't affect the intestine. They used a random forest machine learning algorithm. So there we go. One of the earlier machine learning algorithms we've been talking about on our podcast today, the live one, and identified other chemicals. They found another 20 that they were able to properly identify with some post hoc testing. And then one of these is an herbicide, propazamide, which increased inflammation. And they were able to go into that and find that it works by inhibiting the AHR, the aryl hydrocarbon receptor, which then downstream inhibits the NF-kappa BC and EBP beta signaling in T cells and DCs, which worsens disease. And that worsens TNBS colitis in mice. So aryl hydrocarbons are really important microbiota-mediated signaling pathway that's important in intestinal inflammation. And so they're able to find that a herbicide did this using a combination of zebrafish screens, which are a really cool model organism that I'm also biased towards, and machine learning. And then next up in this little cohort, which I'll then pause and hand back to you after this one, was Viola Lisica from University of Chicago looked at how IgA controls enteric virus colonization and improve intestinal immune homeostasis. So if you knock out IgA in mice, you don't see a lot to begin with until you push on the system. Then you see a ton of stuff, particularly in the gut. So IgA deficient mice have expansion of CD8 alpha beta T cells, but not in germ-free mice. So there's that gut component. And there's no difference in microbial composition with or without IgA. And so they were trying to figure out what was causing this expansion in the cells in these IgA deficient hosts. And so they did antivirals, a broad spectrum antiviral, and it removed the expansion. So they did a combination of lamivudine, ribavirin, and acyclovir. And so then they were saying, wow, the viral compartment seems to be driving this IgA expansion. And they found that murine astrovirus causes the expansion in the small intestine, and so did a norovirus. And the expansion is a compensatory mechanism for the infection. So usually what they're showing is the IgA is coating these viral particles, covering them, letting them pass through your body unharmed, right? And getting out of there, not letting them do their thing. And so in the absence of that, you have more activation and you have to have an expansion to protect the host. And so that's a CD8 expansion. And so that was pretty cool to see, getting some more information on what IgA is doing in the gut, which we know it's super important, but it's also really hard to do experiments on for a variety of reasons. All right. So what was your first one?
Well, I did a little bit of session hopping, and I'm going to talk about the second session I joined, which was titled Recent Breakthroughs and Emerging Therapeutic Strategies for Chronic Inflammatory and Fibrotic Diseases. And I got there for the second half, but I thought it was really good. So the first talk that I really liked was unsurprisingly talking about T-Rex. And this was by Tanya Velez from University of Virginia. And I thought it was very interesting because it has been established that there's a particular population of Tregs that are expressing GATA3 that are found in tissues, and I would say they're often associated with kind of tissue tolerance. And because we see them in tissues, we think, well, maybe these are the regulatory T cells that are keeping these tissues in check. Then this GATA3 is required for their survival in this environment. Because also something that we see in some other cells that are associated with tissue, like ILC2. So in this study, she shows that in patients with inflammatory lung interstitial lung disease, I think it's called, they it's a fairly rare disease, but it can be very fatal. So this, I think, is a very interesting thing to look at. And they show that so ILD patients show germinal centers in their lungs, and part of the pathology is mediated by antibodies. And basically, she looked at kind of regulatory T cells, and she does see that in patients, both in patients and in mouse models, there is an increase of GATA3 positive Tregs in these lungs, which might suggest that maybe this is the way Tregs are. They're that's how they're doing the job. This is like the subset of Tregs that are trying to help with inflammation. And so in a case when you have a mouse model in which you're inducing the disease, you see that this peak of of both total Tregs and GATA3 positive Tregs around 2 weeks after the start of the disease. And that depleting the Tregs, if you deplete all Tregs using a diphtheria model, basically that kills the mice. So some mice could potentially recover from it, but not if you get rid of the Treg. But I thought what was very interesting is that actually, if you deplete GATA3 Tregs only, that actually helps. So the presence of those Tregs, of those GATA3 positive Tregs, actually seems to be detrimental to the disease kind of resolution. So I thought it was very interesting. I'm definitely gonna keep an eye on where her future research is going on this area. And then after her was uh, I think I have always a fun pronouncing Cheesy Tang with a Q, and she is a uh, her lab and her work. She's been working on Treg and engineering Tregs and therapeutic Tregs for many years. At UCSF at the University of California, San Francisco. She's a authority on Tregs for therapy. And so she was talking about engineering next generation Tregs for type 1 diabetes. And what I thought was very interesting is that basically she gave some background about what we understand, like what has been our approach or her lab's approach. I know she's been kind of trailblazing this to using Tregs for protecting against type 1 diabetes, and which are the mouse models that help study this disease and the potential for Tregs for helping. And one of the things that she discusses is the results of a phase 1 and 2 trial in which people get expanded Tregs. So Tregs are taken out of the body, they're expanded ex vivo, and then they're infused. And the idea is that if you infuse enough Tregs, you might be able to kind of help protect pancreatic cells from destruction in type 1 diabetes. And what she says is that although Tregs can be expanded in vitro to really large amounts, billions of cells, they can be infused and people tolerate them well, doesn't seem like this Tregs really persists very long, especially like you see a very large crash in the first couple of weeks, and you see some persistent but of fairly low numbers. And this is something that I think also as happens in other types of cell therapy, and especially T cell therapy. And one of the questions is whether this might be due to the starvation that the cells have suffer of IL-2, which is used in high amounts of IL-2 are used during culture of Tregs. And then when you infuse it on the patient, there's only so much IL-2 you can actually give a patient without killing this person, because it does activate other conventional T cells. So this is a well-known effect of IL-2 infusion is that you have a large kind of inflammatory response, and it can be very dangerous. So her lab embarked into trying to find some way of modifying these Tregs in order to give them a better source of IL-2 that is not injecting IL-2 to the patient. And this is where they tapped into these orthogonal IL-2s, which are actually a work from the lab of Christopher Garcia. He's very well known for these mutants and these modified cytokines. And basically, the idea is that you can generate a mutated version of IL-2 that only binds to a receptor, and then you have that cell expressing both the cytokine and the receptor. So then it's kind of driving its own activation, its own how do you say, feeding in a way. And so that was very interesting. They also show that if you actually tether the cytokine to the receptor, then you kind of guarantee an autocrine stimulation. And actually, that seems to be better. They look at different variations of this autocrine receptor cytomucin combination. But in general, she showed that there's a lot of promise in trying to use these approaches that increase kind of specificity, because you only want to feed the Tregs, but not everything else. And so you can also play around with maybe even if you have a cytokine that has lower kind of binding affinity or lower in signaling, as long as you are the only cell expressing that receptor or you are tethering to a cell, then this can be good enough for your Tregs to really grow to the amounts that you need. So very interesting. This is a super dynamic field, I think. And last in that session was actually our guest at the live session, then Joe Viney from Seismic Therapeutic, because she was talking about some of Seismic's pipelines in which they're developing molecules for therapy for autoimmunity. And I thought it was really, really interesting story, because she talked a lot about, well, if we know that they're, inhibitory receptors, they're bad for cancer, but they might be good for autoimmunity. So why don't we kind of focus on those? And so they developed this PD-1 agonizing antibody, and she went through the development on how do you find what are the constraints that you have when you're designing, when you're choosing this antibody. They also actually use some machine learning to kind of predict from a broad amount of monoclonal antibody candidates, which would be the ones that are binding to the places you want them to bind within the PD-1 receptor. Because when you have an agonist antibody, you don't want to block the function, right? You want to be actually, in a way, exacerbate or synergize with the natural ligand. And so it's very interesting. And I thought what's also super interesting is that in this case, they're having this dual cell, what they call dual cell bidirectional agonist, because on the one hand, they have a monoclonal antibody that binds to the PD-1, but then the monoclonal antibody has a FC portion that can bind to an FC receptor. And then if you bind it to the right FC receptor, then in this case, FC gamma receptor 2 beta, then you can also induce kind of a dampening of the immune response on the antigen presenting cell as expressing the receptor. So again, it was a lot of like this modulation of the binding of the antibody, of the constant domain of the antibody to the right receptor in order to not only dampen the immune response on the T cell or inhibit the T cell, but also in a way inhibit the APC itself. So you have like a double punch approach. I thought was super cool. And the way that she explained how the kind of analysis that they did, I thought it was super interesting. Very, very cool science in general. And then by the end of the day, last talk I thought was so good. I just want to highlight the talk by Stephanie Eisenbarth from Northwestern University. She won an award for the Lansfield Mid Career Achievement Award. And I thought her talk was so good. So she started like in a way, it was kind of an anthology of her lab's work and of her her research as an independent in her independent lab, and she started talking about how kind of a chance discovery in which they had this mouse model that had what they thought was a defect in the inflammasome pathway. They published it, and then it turns out it was actually not that. That not was that the gene that they thought that was generating the phenotype was a different gene called DOC8. And this discovery, so she kind of pivoted towards studying DOC8, and one of the things is that DOC8 deficiency, or this SNP on DOC8 was generating was in hyper IgE phenotype, which then also associated to some observations in humans. And this really got her kind of down a rabbit hole of allergy and how can this particular mutation generate this allergenic phenotype in mice, and then how this associates with human. So a 1 hour talk was really good. But I think the most important thing that she showed is that by kind of following the science in the way it appeared to her, she actually discovered that most of the mouse strains that we're using, except Black 6, have a certain predisposition for allergy. And this is something that Black 6 don't have, and the rest of the mice do. And they use this to understand where is this allergy coming from. And it turns out that one of the things they really pinpointed is production of IL-13 by T follicular helper cells. So this is like this deficiency is from T cells, and is the follicular helper cells that have this DPEP protein, DPEP1, that is a carboxylase a protein that one of the things that it catabolizes is leukotrienes. And so overexpression, or kind of more efficient expression by Black 6 mice, actually ends up protecting them from internalization of allergens through the gut epithelium. And this is why Black 6 mice don't get these allergic reactions that almost all of the other mouse strains do. And this actually also seems to correlate with human phenotype, in which the homologous protein in humans also has a similar sequence that presumably also explains, at least partly, where these very high anaphylactic responses come from in humans. So, and this, as I said, is mediated by IL-13 expression that allows for high affinity IgE clones to be generated that are mediate much more stronger mast cell activation. Of course, I'm here saying a very short kind of version because I don't want to go too long, but the way she explained the whole story was awesome. And she was such a nice speaker, talking about her team and her. She started at Yale. She did most of her career at Yale. Then she recently moved to Northwestern here in Chicago. And I thought she was such a nice explanation of how you get a career going, and all the unexpected things that came throughout this time. And I thought it was really, really good. Um, so that was the last talk I wanted to talk about.
All right. Well, I had a couple more I went to as well throughout the day. For my big symposium, I went to see Dr. Kim Rathmel, the new NCI director. who actually was one of my mentors when she helped lead the MD PhD program when I was a student at UNC. And so it was great to catch up with her and then see what she was talking about with the role of how we can end cancer as we know it. So she talked some for faculty about the funding environment and did a pretty good deep dive there. But then she also made the point that, now everyone wants to be an immunologist because of the role of immunotherapy in treating cancer and really driving bottoms up research now. So, really understanding core principles of the immune system and how to use that to drive new therapies. And she also mentioned, guess what, the rise of AI and used an example of the rise in colon cancer potentially being linked to a single bacteria that's becoming more common. She talked about the National Cancer Plan and that roadmap, and then focused on support that they were providing for young investigators throughout the NIH, which is the main funding agency in the United States. And then for my last set of talks, I went to mechanisms mediating inflammation in the afternoon. This was more colonic than I was planning on it being, but there were a couple really interesting talks from there. The first one was from Felipe Teixeira Lima at the University of Florida, called deletion of MOF in CD4 T cells results in mitochondrial dysfunction, Th17 polarization, and chronic inflammation. MOF stands for male's absence of the first. And I just have a pet peeve of this name because that's just not a helpful name. Um, it's a histone acyltransferase and regulates respiration in mitochondria. And that's what was known. What they showed was MOF knockout in CD4 cells alters their mitochondrial morphology, not surprising, and they have impaired oxidative phosphorylation in all steps, and that results in more ATP production as a compensatory mechanism. And that there's something going on because these mice develop spontaneous colitis, but they haven't finished the story yet about why they're developing spontaneous colitis. So leaving that there that we don't understand that yet, work ongoing. They did a mitochondrial antioxidant that restores the function of the mitochondria, and that reduces the colitis. There's something about immunometabolism going on here with mitochondrial functions and disruptions in that leads to spontaneous colitis. The next one up I had was Sarah McNear from Case Western, which is estrogen signaling through ERA exacerbates intestinal inflammation selectivity in males. Sorry, ERB. So what they showed was that there's ERA, my bad. T cell transfer colitis is the model they used. Well known, for being a T cell mediated. You take T cells from a healthy mouse and put them into a rag knockout and they get colitis. And they showed a pretty profound selectivity in males. So they had an ERB knockout, so estrogen receptor beta knockout. And these mice had exacerbated colitis, but only in male transfer versus female. And it wasn't a result of the CD4 cells, and they aren't sure exactly what's going on yet, but they were noticing increased secretion of IL-17 and GM-CSF that was leading to this. Now, as transcription factors, there's a lot they can do here. And so a thing to note is that in addition to these estrogen receptors binding to their, promoter region on itself and activating, things specifically regulated by the estrogen receptor spot, it also will bind to other promoter regions and have other activation or inhibition of other genes. And so they're wondering if that's what's going on here. And then lastly, we have Christina Wilkinson from the NIEHS. She was looking at the role on her talk to the title of GM-CSF regulates lung homeostasis by promoting development of CD301 beta positive resident dendritic cells that induce regulatory T cells. So she uses a house dust extract / ovalbumin model. So house dust extract is this exactly what it sounds like, extract from house dust that's used as a model of allergy, but you then sensitize with ovalbumin. And so she was talking about how there are multiple different types of canonical DCs, and she was looking at classical DC2s, and then found there was heterogeneous development under inflammatory conditions. And they did that by, guess what? Single cell RNA-seq, our favorite thing. And she showed that this inflammatory drive that's leading to different differentiation had effect on downstream Treg differentiation. The Treg differentiation is induced by the subset of CD301 beta positive DC2s. And so inflammation leads to this 301 positive state. Inflammation leads to this 301 positive state, which then leads to a Treg program being activated. And they're still figuring that out. But one of the things involved was TGF beta. And so that ends my day as well.
Fun. It was a very interesting day. But man, it's so hard to choose a session over another, huh?
It's hard to make choices, but we all have to sometimes.
All right. So that brings us to the end of our show and the end for day 2 at Immunology 2024. Don't forget to follow us on X at ImmunoPodcast to find out what we're up to at the meeting, and visit us at the Immunology Podcast booth on the exhibit floor when you can win a prize and find out how you can be featured in a future episode of the podcast. Come back here tomorrow for another episode, recapping day 3 of the meeting. See you then.